trying to get cells to make a lot of soluble protein and they're refusing, perhaps you should be using a fusion. So fusion partners or fusion proteins are these little proteins, things like SUMO and GST and MBP, NUS-A, TRX, that we can stick onto the ends of a protein that we want cells to make for us. And this can kind of trick the cells into making our protein because they really like making that other protein. And they don't just like making it, but it actually folds up really nicely and it stays soluble. So it can help your protein stay soluble and help your protein fold as your protein is getting made. And so today I want to tell you more about these fusion partners. Some of them, like GST and MBP, they actually can be used to purify them as well. They can serve as affinity tags. We can use like glutathione or amylose resin to purify them. But they have some problems, like they dimerize and things like this. I prefer SUMO. I'll tell you a little bit more about all of these, but SUMO, it also has this cool thing where it has this specific protease that'll cut it off. And so much more about these and other topics, so let's dive in. If it works according to plan, recombinant protein expression is awesome. We can take the genetic instructions for making a protein and stick them into a vector. So this is basically a piece of DNA that's going to act as a vehicle for getting those genetic instructions into cells. And so we could stick these into bacterial cells or yeast cells or mammalian cells or insect cells. And these cells, basically, because the genetic instructions are going to be written in this universal code of the nucleic acids of DNA and RNA, these cells are going to be able to read those instructions and piece together the amino acids, telling them which these protein letters to add in what order. And voila, you end up with a protein. But sometimes things don't go according to plan, because even though these cells are able to read the recipe, they might not want to make the protein. You might have problems with expression, so actually getting that protein made. You might have problems with the stability of the protein, and a big problem often is solubility, where basically the protein is going to come out of solution. It's going to kind of clump up and aggregate. This can collect in what we call these inclusion bodies, where basically you have these clumps of misfolded proteins and things. And there are ways where you can actually purify out those clumps and try to renature or refold the proteins. But ideally, typically, you want a nice soluble protein. And so when we talk about something being soluble, it has this full water coat. And so each of these copies of the protein is going to have this coat of water. And so this is what we want. But sometimes we end up with this. And we end up with, when we go to purify out our protein, we break open those cells and now we want to go purify them, such as with chromatography, which I'll talk more about later. Basically, when we do this, we go and we see, okay, well, I don't see the protein. And so this could be either because your protein wasn't expressed, or it could be because it was expressed, but it was going into those inclusion bodies. And so much more on this in other posts, but basically if it goes into the inclusion body, when you centrifuge the lysate, so basically you break open the cells, we call this lysis, then you spin down the cells so that the heavy stuff is going to kind of sink down. And so this is going to be all like those membrane bits and stuff from the broken open cell. But this is also where insoluble things are going to go, such as those inclusion bodies. So your protein might end up in here. And so if you run an SDS page gel on these different fractions, so on the pellet and the supernatant, so the liquid above it, this can help show you if your protein was made. If you do like a Western blot or something to use antibodies to detect the protein, you could see potentially, well, was it made, but it ended up in these inclusion bodies, or was it not made at all? And so both of these issues can be helped with these fusion partners. And so today I want to tell you more about these fusion partners. And if you want to know more about other types of protein expression or solubility issues, I'll point you to my post on that. There are multiple strategies that you could take, and today I want to focus on the fusion partners. And so the fusion partner is going to be a small little protein that the cells really like to make. So they like to make it, and it's going to fold really easily, and it's going to stay really soluble. And so this is going to kind of trick the cells into making your protein, because you like tag on that fusion partner to the front of the instructions for making your protein. So it starts with the instructions for making the fusion partner, And then it keeps going because there's not a stop codon in between. So basically there's no stop signal here. There's a stop signal at the end of your protein, but not at the end of the fusion partner. And so when the cells go to make the protein, they're going to make the fusion partner. They're really excited because they like to make this one. So this could be like a SUMO or GST or an MBP or an SA or TRX. And we'll talk about a few of these examples. And then it will go and it'll keep making your protein. So on its own, your protein might not express very well. It might not get made very well. But because it's attached to this fusion partner, well, now this can help with the expression. We'll also talk about how you usually stick like a protease recognition site in between the fusion partner and your protein. So this is going to be a sequence that is specifically recognized by an endoprotease, so by a protein cutter. That cuts in the middle of a protein, and it'll cut at the specific sequence. And so we can use things like TEV, SUMO protease. It will actually recognize like the SUMO tag. We have thrombin, various other sites that we can introduce, and this is going to help us cut our protein off. And this can be really, really important with these fusion partners because unlike just having a small his tag on the end, a small little tag to help you with affinity purification, which we'll talk about, these are actually whole proteins. And so these can get kind of big, and we'll discuss the relative sizes of some of them a little later. But basically, you often are going to want to cut it off later. But beware that when you cut it off, your protein might now go back to clumping up. And so one of the ways that the fusion partner was helping was actually not just with the expression, but with keeping your protein happy and soluble. And so remember that when I said when something's soluble, each of the copies has a full water coat. And why this happens is because these proteins fold up in a way that puts their hydrophilic, so their water-loved parts on the outside where they can hang out with water, and then their hydrophobic things, so the things that water avoids, they kind of hide these into the center of the protein. The problem is that proteins have these like 3D shapes where they're going to hide those hydrophobic parts in the middle, but those hydrophobic parts are not always going to be coming out at the same time. And so as the protein is getting made, as it's getting translated, basically this amino acid chain, this nascent chain is coming out of the ribosome. And as it's coming out, it has to fold. And the way it's folding, a key driving force of this folding is going to be that hydrophobic effect. So those hydrophobic parts being clustered together. But if things don't go well, those hydrophobic parts, well, they can be out there just kind of like hanging out and they can't find their partner. Maybe it hasn't come out yet. Maybe they misfold a little. And so this can cause the protein to misfold. And if it misfolds, those hydrophobic parts, they become exposed to the water, and the water doesn't want to hang out with them. And so what's going to happen is the protein is going to kind of like clump up with other proteins, other hydrophobic things. You get these problems like these inclusion bodies, and you can have the cells where they'll either just kind of like sequester those proteins, or the cells might just like degrade those like misfolded proteins. So in either case, you're going to have some issues. Now, why doesn't this just happen in all cells with all proteins? Well, part of it is that sometimes proteins that need help folding, they actually get help folding in cells with the help of chaperones. And so chaperones are kind of these proteins and big like protein complex type things that actually kind of help the protein fold. And so they can do this by kind of when the nascent chain is coming out. So when the protein is coming out as it's getting made, they can kind of hide those parts that are hydrophobic until like the partner for the hydrophobic part comes out that hasn't come out of the ribosome yet, because the ribosome, well, it's still working. And so when this part needs to be bound to this part, sometimes what happens is that this part up here is going to kind of get shielded by the chaperone until the second part comes out, until its partner comes out and it can get there. And so chaperone proteins can often play this role. And it turns out that some of the fusion proteins can actually serve as a sort of chaperone role, where they can kind of have these hydrophobic patches that can kind of help the protein as it's coming out, kind of shield it from the water and protect those parts, help the protein fold properly. Some of them can also kind of recruit other recruit the actual chaperone machinery to try to help out. So those are a couple of ways that this can help this protein fold properly. And as this is all going on, these chaperones or these fusion partners can also protect these proteins from the proteasome, from like degradation and things like this. So you can think of this fusion partner as often playing this role as kind of protecting the protein as it's getting made and helping it fold properly, as well as kind of tricking the cells into making it. And then because it's really soluble, it's going to help the protein stay soluble, even if your protein part isn't that great in terms of solubility. It's going to be attached to this really soluble thing that's going to kind of make it so that the little protein part on the end can't all clump up together. So fusion partners can be really, really helpful for a variety of various purposes, and different ones might work in somewhat different ways. But these are some of the key ways that they work. They can also kind of move proteins into parts of the cells where there's less proteases. There's a variety of different things, and I'll point you to a review article that's really good at explaining these. This article also has some really good information about the sizes of these different tags. And so I talk about these like little proteins on the end, but these can actually get pretty big. So the one that I've used the most is this SUMO tag, this small ubiquitin modified, which is this protein. And so it's kind of like ubiquitin, and it actually gets added onto proteins in a manner similar to ubiquitin. It's this modification that can get added onto proteins naturally, post-translationally after the protein's made. But in this case, we're actually getting it made in having it attached to our protein as our protein is getting made. And this is going to help it by serving as this fusion partner. But it's also going to add a lot of bulk. And so in this case, the SUMO is about 100 amino acids. It's about 12 kilodaltons. And so you can see if you run an SDS page gel, you can see that if you cleave the tag, then you can get a reduction in the size of your band, and you see the appearance of a smaller band representing that portion of the protein that was cut off. It'll be pretty low down because it's only 12 kilodaltons, and then you also see like the protease that you added. Speaking of which, with SUMO, you can use like a SUMO specific protease that's actually going to recognize the shape of the SUMO protein rather than a specific sequence, or you can introduce like a cleavage site for another protease. And in these cases, it's really important that you remove that tag often, because these tags are these big bulky things as opposed to just a little affinity tag. And so another problem with some of these is like GST and MBP and stuff. These have a tendency to form to like oligomerize, and so they can actually like form dimers and things like this, which can make your protein then like clump up, and it can cause it can run weird on gels. So just a warning about that. So I like the SUMO tag. So on a technical note, the SUMO protease, as I talked about, it's really awesome in that it recognizes like the shape of SUMO and it cuts next to it. And it does this in like this scarless way so that you're left with the native N terminus of your protein. So you don't have to worry about like extra linker left on. But your cells actually, like human cells and like other types of eukaryotic cells, they have SUMO proteases. And so if you were to try to use the natural SUMO tag in the eukaryotic cell expression system, something like mammalian cells or insect cells, well, then those cells could actually cut the tag off. And so this tag works great in bacteria, but if you want to use it in eukaryotic cells, here you have to actually modify the SUMO. So there's like a different version of SUMO, like a SUMO star, where basically it makes it so that this tag doesn't get recognized and cut off by those proteases in our cells. So by those SUMO proteases that are present in cells, it won't cut the tag off. But then you have to use a cut sequence for something like TEV. Alternatively, you can use like a modified version of the SUMO protease. So instead of using ULP1 or whatever you would normally use to cut off the SUMO tag, you can use like a modified version of SUMO protease that is still going to recognize the shape, but it's going to only cut this modified version of SUMO as opposed to the naturally occurring version of SUMO. So it's kind of like mutate both parts so that now they work together again, but they don't work with like the natural stuff. But the way that I've done it in the past is just by inserting a TEV tag, a TEV cleavage site in between the SUMO and my protein. And it's still just a short little sequence, so it's not going to leave much left over. And so in this case, you can have like a strep SUMO TEV, where there be a TEV site between your protein and the fusion partner. And so that's what you see in this example of a purification that I did in my PhD work, where I had a strep SUMO TEV tag, and then I used that TEV site to cut the protein off. And I used this modified version of SUMO because I was working in these insect cells, which would have cut the tag off inside the cells and cause problems if it was the natural SUMO version. So I'll post a link to an article on this as well. Another thing that's great about the SUMO tag, though, is that you have that SUMO specific protease that you can use. And because it's recognizing the shape, it doesn't care as much about the linker. Whereas with some of these other tags, the linker can be really important in terms of where the cleavage site is placed, and that it can be hard for the protease to actually find the sequence, find that cleavage site. And so you might need to play around with the length of the linker between your protein and the fusion protein. And the fusion protein might actually like hide the cleavage site. And so that could be an issue if you're having problems with the cleavage. You might want to try playing around with that linker. Okay, so that's just a technical note. So let's look at a couple of these other ones. So I like SUMO for a variety of reasons, but one of the things about SUMO that isn't good is that it can't act alone as an affinity tag, whereas some of the fusion partners can act as an affinity tag as well as a solubility helper. And so a couple of these examples are GST and MBP. So when we're talking about affinity tags, I'm talking about something that we could use for affinity chromatography. So much more on chromatography in other posts, but basically we have these columns that are filled with these little beads called resin. And these resin have different properties, and proteins have different properties. So the proteins are going to interact different proteins will interact differently with different resins, and therefore they're going to flow differently through these columns. And this is going to allow us to separate the proteins based on various properties, like their charge or their size, or in the case of affinity chromatography, based on the presence of something like an affinity tag, which is something that our protein has, but other proteins don't. And so if we have a binding partner for this affinity tag attached to that resin, we can get our protein to bind, wash everything off, and then elute or like kind of push off and compete off our protein. So a couple common examples are like a his tag or a strep tag. And so these are really great because they're really short. And so they're not going to basically interfere with our protein typically. And so we wouldn't even have to worry most of the time about cutting that tag off. In the case of one of these fusion partners, however, you have this big bulky protein. And so you're going to want to cut it off typically. So with the SUMO tag, this is just going to act as a fusion part. It's going to act as like a solubility helper, but it's not going to act as an affinity tag. So if we want to affinity purify a protein that has a SUMO tag, then we stick on the end of it an affinity tag. So for example, we can do a strep SUMO tag, where we have a strep tag on the end of a SUMO tag attached to a protein. And this is going to enable us to then purify using that strep tag and have the solubility and the expression help from that SUMO protein, getting us to make our protein, and then we can cut this whole thing off. So cut the affinity tag and the fusion partner off at the same time. In the case of some of these fusion partners like GST and MBP, they can actually act directly as affinity tags. So GST is glutathione S transferase. So glutathione is this little like antioxidant molecule that our cells use in order to kind of help maintain a reducing environment. So make it so that cysteines and proteins, that they don't form like unwanted cross links where these cysteines cross like hook up to one another. And so much more on this in my post on cysteine. But glutathione is kind of like what your cells use instead of what we use in the lab, like DTT. Glutathione is kind of like a cellular version of that, or DTT is like the lab version of it. Basically, these are used to help maintain a reducing environment and prevent these unwanted cysteine crosslinks from forming. Glutathione as transferase is an enzyme that actually binds to glutathione. And so if you have glutathione as transferase on the end of your protein, as one of these fusion proteins, so it's helping with the solubility, it's helping with the expression, and it also has this added benefit of, oh, hey, it can bind to glutathione. And so if we stick glutathione onto agarose resin, so we have these basically boring beads, but then we conjugate or we permanently attach to them glutathione. Well, now our protein, the affinity tag or the fusion partner attached to our protein, so this GST, it binds to glutathione, and it's going to bind to the glutathione, whether that glutathione is free or whether that glutathione is bound to the column, bound to the beads. And so when you take your protein and you flow it on through this resin that has this glutathione, well, now your protein is going to bind to the beads through the fusion partner, through that GST. And then once everything else was washed off, we can then push off, like elute our protein off by adding free glutathione, which is going to swap out for our protein. The fusion partner is going to swap that out for the glutathione that's on the column, because you basically flood it with this free stuff. And so now your protein is going to come off. We can use a similar strategy to purify MBP tag proteins. So MBP is maltose binding protein. It binds to some sugars, including amylose and maltose. And so typically you use this like amylose resin, and then you compete it off with maltose. And then again, what nice about these is that they have the affinity and the like solubility and all that stuff built into this one protein. For other fusion proteins, we might need to add an affinity tag onto the end of it if we want to be able to use it as an affinity, if we want to be able to like affinity purify these proteins. But with GST, I'm not a fan of GST, like personally, just for using it, because I've had problems with dimerization and things like this, as well as sort of like degradation on gels and things. So, but a lot of people use it and seem to have good success. And so you might actually need to try different fusion partners. If you're having trouble with the expression, you can try different fusion partners. You can try changing the linker. And these are just a couple of strategies using fusion partners, but then there's a ton of other things that you could do if you're having problems with expression or purification, and much more on that in my troubleshooting post. And things like codon optimization, using different strains of bacteria for bacterial expression, all sorts of things. You can also play around with where you put the tag. In addition to the linker, you can put the tag either like on the N terminus, so the beginning of your protein, or the C terminus, the end of the protein. So in either case, it might be able to help it stay soluble once it's already formed. But typically you want to put these tags on the N terminus because you want it to be made before your protein is getting made, so that it tricks the cells into making your protein, and then it helps your protein get made and folded. And then it helps it stay soluble, rather than just helping it stay soluble after the protein is already made. But some proteins, they have things on their like N terminus that would make it so that you wouldn't want to actually stick a fusion partner there. So it's going to take some troubleshooting and optimizing and finding what's best for the protein that you're working with. But fusion partners can be a really helpful tool when you are trying to purify a protein and these cells are like, no, I don't want to make it, or no, I'm not going to make it right. And this can also often be an issue with like bacterial expression. Because with bacteria, if you're trying to like express a human protein, bacteria aren't going to have all the same machinery that our cells have. And so they're not going to have the same chaperones. They're not going to have the same like post-translational modifications and things. So the cells are going to kind of have to have a harder time making those proteins. And so a fusion partner is something that can kind of help help get these cells to make your protein. One more thing when it comes to solubility, especially with overexpression in bacteria, sometimes you're just asking the cells to do too much too fast. And so with these overexpression, you're basically saying, make this, make this, make this. And this isn't giving time for the proteins to get made and folded properly. And so sometimes if you actually kind of like don't overexpress it as much, you can lower like the induction, the IPTG, lower that concentration. You can also try lowering the temperature that you're expressing at, kind of slowing things down so that the protein has a little more time to fold, as well as lowering the total amount of protein in there that is then going to be available to help help clump things up. And so basically there's other strategies you can take to try to help solubilize things in bacteria, and you might just need to slow things down and lower the levels of things. So much more on that in my other post on troubleshooting expression I'll link to. But if that doesn't work, you might need to turn to a different type of cell lines, such as mammalian cell lines, CHO cells, or freestyle or XB cells, or insect cells, baculovirus things, yeast cells, various other expression systems. But with any of these systems, you can use fusion tags, but the best ones for different systems might vary. And so, yeah, so I would check out that review article that I will link to, and hope this helps, and hope you don't have to go down this a ton of troubleshooting with your expression. So good luck.